And so a lot of the work that came about with the germ theory of disease, some of it was by Lister. Name sound familiar? Listerine, who showed that wound cleaning reduces childbirth fever or puerperal fever. So obstetricians at the time, were, the dirtier your equipment and your gown was, the more prestige you had. But in actuality, it was really a dangerous thing because you were introducing germs during childbirth. So Lister would use clean equipment, clean gowns, and reduce the incidence of childbirth fever. And also showed that wound cleaning with a phenol solution would promote healthier or faster healing and fewer infections. And so, ever heard of phenol before? Phenol is a disinfectant. We'll learn more about disinfectants later on in this class. Pasteur developed aseptic techniques used in the laboratory for manipulating bacteria that you're learning now. And he disproved spontaneous generation, but he also developed pasteurization. Did a lot of other things too. But Robert Koch developed he had a lot of contributions in microbiology. His most famous ones are Koch's postulates. The postulates are a set of criteria that explain, or not explain, but help to determine which organism is responsible for a particular disease. So it associates a particular microbe with a particular disease. So it's a demonstration that a disease is caused by a specific microorganism. And a little bit earlier in this whole game plan was Jenner, and Jenner showed that he developed the first vaccination. And he didn't really know what he was doing, but at the molecular level and at the immunology level, but he made some clear observations, and he put those observations to work. He noticed that smallpox was rampant at his time. This was in the late 1700s. And he noticed that two observations. One, that cows get a similar disease, but they don't die. Two, that people working with the cattle don't get smallpox. So he said maybe there's an association between the disease that cattle get and it protects people somehow from the disease that humans get, because they're both similar diseases, cowpox and smallpox. So what he did was he took samples from the pustules of diseased cattle and injected them, a crude injection, just like breaking the skin, on people with that sample. So they would take the sample from the cow and break the skin and kind of inject it into the person. The person would develop a real mild form of pox, but they would not get, and that protected them from smallpox. They would not get smallpox. So it was the first immunization. So we talked about this last time. What was the thing that was going on at this? What scientific thinking did we develop at this time?
Immunization?
No. In general, I'm talking about what was the thought process that was going on. They developed a very important set of ideas. Just say it. Doesn't care. I don't care if you're wrong. Yeah, that was all going on, but before that, just the scientific method. Yeah, the scientific method. Just the whole idea behind science and how to make new discoveries, and that's really what the driving process to all of these advances, is the scientific method. Robert Koch, I said before, he did a lot of interesting stuff. He developed organic dyes for staining microbes. He developed methods to culture bacteria. He developed agar. What is agar? Agar is that hard stuff on the plate that you were plating on. What's that made out of? What? Doesn't matter. Carbohydrate. It's a carbohydrate. But what other carbohydrates were being used at the time? Before agar came around? Starch. You know, starch comes from plants. Gelatin was also used. What's wrong with starch? What's wrong with gelatin? Why aren't those good substrates for growing bacteria?
Don't they have their own bacteria that come with it too?
Yeah, bacteria eat it. So they eat the material that you're trying to grow them on. They don't eat the agar, which is another type of carbohydrate. Why not? Because it comes from a different environment. It comes from algae. So there's not very many bacteria that eat algae. Where would you find bacteria that eat algae? In the sea. Right. So if you want to grow marine bacteria, you got to use something else. Koch's postulates. How to prove a certain organism causes a particular disease. Here they are. 1. You have to see the same pathogen present in every case of the disease. You got to see it. So you got to be able to look at it and say, oh yeah, that looks like it's the same bug. So they're looking at a cow and they're showing that the bacteria is present in the lesion. And then you have to isolate the bacteria in pure culture. You got to get it in the laboratory, away from the animal, and grow it in the laboratory in pure form. Okay, and you're learning how to do those techniques now with your sterile technique and with your street plate method. Then you got to grow it, grow lots of it, and then you have to reintroduce it into a fresh, clean animal, with an undiseased animal. And it causes the same disease you saw in the first animal, the one you got it from. That's the third one. The fourth one is you have to re-isolate the organism from the second diseased animal, the one you introduced it to, and show it to be the same as the first one that you isolated. So it's pretty logical, you know. You find a diseased organism, you take the diseased bacteria, you grow it in the lab, you put it into a new host. That host gets sick, you show that and re-isolate the organism and show that it's the same as the first one. Yeah.
So when you're saying Koch's postulate, you're talking about the scientific method, but early version of it? It's not the scientific method. Well, he probably used the scientific method to develop the postulates, but they're two different things. The scientific method you can apply to anything. This you can apply only to showing a disease causes a bacteria causes a certain disease. I already explained Jenner's smallpox vaccine. Ehrlich. Robert Ehrlich. He was around the turn of the century, 20th century. He developed the first drug that was active against syphilis, and which was rampant at the time. And syphilis has been rampant throughout history. So this was a great find that he found a drug that was active that people could take that would cure them of syphilis. The way he found it was a very systematic approach that pharmaceutical companies today still use. It's very time-consuming and it's very laborious, but what he did was, he's a chemist. So he knew this organism, the causative agent of syphilis, was sensitive to certain
classes of chemicals that these chemicals would kill. But you can't inject these chemicals in people because it'll kill the people too, right? So that's the key, to find something that'll kill your target organism but not the host. So what he did was he took this drug, this chemical, and he made one chemical modification to it. Added a methyl group, or took off a methyl group, or added a double bond here, or removed a double bond there. It's a large process. It's a large molecule, so there's a lot of things you can do to it. Made one change to it, gave it a number. Made another change to it, gave it another number. Made another change to it, gave it another number. So he developed a library of chemicals, and he must have developed almost 1,000 of different chemicals that were derived from the one main compound that hurt the bacteria. And then he tested each one of these for activity against the microbe to see if it still retained its activity in killing the microbe, and also for its toxicity, whether or not it would hurt a host. And so he screened all of these different compounds. Number 606, called arsphenamine, which was a related arsenic compound to the original compound, was active against the bacteria in question. And it was also had a low toxicity. So that's how we found it. So it's a very laborious approach to identifying drugs, or synthesizing, I should say, drugs in a laboratory that are active against their bacterial targets.
How long did it take him?
I don't know, 600 of these things? I would say, I don't know exactly, but over a year.
And was syphilis always like sexually transmitted?
Generally, yeah.
Just,
It could also be transmitted in childbirth. That's one reason why you still give silver nitrate to newborns in their eyes, because they get these syphilis infections in the eyes, which cause blindness. And so it's a cheap method to guard against disease. The silver nitrate is. That's why it's routinely done. Alexander Fleming was the first to develop the antibiotic. Why don't we call arsphenamine, or Ehrlich's compound, why don't we call that an antibiotic? It's not an antibiotic. Why don't we call that an antibiotic?
because syphilis is a viral infection?
No, it's bacterial. It's because it's a compound that's made in the laboratory. Antibiotics, by definition, are made by other microorganisms. So Fleming discovered the first antibiotic, which is penicillin. And this was about 25 years, 30 years after Fleming. After Ehrlich. So he noticed that when he was growing bacteria on a plate, agar plate, that his plate was contaminated with a mold. And he saw that in the vicinity of the mold, the bacteria weren't growing. And so he theorized, using the scientific method, developed a theory and an experiment to test that theory. So his theory was that the penicillin, or the penicillium, which is a mold colony, is putting something out into the medium, secreting something that's inhibiting the growth of the bacteria. So he tested that. How would you test that in an experiment?
Isn't that what we did to that?
No. We're looking at growth. We're not looking at inhibiting growth. But that's good. You're still thinking, yeah?
Multiple trials with different bacteria.
Well, how would that test the theory that something is being produced by the mold and put into the medium? You notice that the mold is not touching the bacteria. but it's inhibiting bacteria far away from it, yeah.
Introduce the penicillin to an already established bacteria, like, how long?
Introduce the penicillin how? Kinda, you're on the right track. Anybody else?
You grow them both together?
Well, I don't know, it depends on how you do the experiment. I'm going to tell you, I guess I have one more answer.
Well, couldn't you try doing the experiment again and then introducing the penicillin after the bacteria grows?
The bacteria would already be grown. Right? It would already be grown. You wouldn't see. You would see it stop growing further. Well, here's what he did. He said, well, if they're elaborating something, if they're putting something out in the medium, well then, why don't we grow the penicillin up, filter out the penicillium, filter out the mold, and then just take the supernatant, the soup that they were growing in, without the mold, and see if that affects the growth of the bacteria. And sure enough, it did. So he took some of that soup that the mold was growing in, without the mold now, and he put it onto a plate, tried to grow bacteria. The bacteria would not grow there. So that's how he demonstrated that. He didn't prove it, but it hasn't been disproven yet. We already saw this. That's that. Any questions so far on history? Okay, we're not going to do chapter 2. Let's go right into chapter 3. But you're still responsible for chapter 2. Now, this is good because it starts off in talking about something we did tonight in the lab. Okay, talking about staining. There's different types of dyes you can have. You can have basic dyes or acidic dyes. Now, basic dyes are going to be basic, they'll have a low pH. They'll have a high pH. And acidic dyes are going to be acidic, and they're going to have a low pH. Stains can be simple stains, where usually one component is used in the process. Or you can have differential stains or complex stains where multiple components are used in the process, like the gram stain, like you learned tonight. And remember, the differential stain helps you to see a difference in structure between different organisms. And you can have specialized stains. And specialized stains allow you to see very specialized structures that not all bacteria will have. Like a flagellar stain will help you to see flagella on bacteria that have flagella. Okay, let's look at a basic dye. This molecule is a basic dye molecule because it is positively charged. So basic dyes are positively charged. The part with the charge is called the chromophore. The chromophore is going to interact with the charges, the opposite charges in the cell. So that's why it sticks, or that's why it is attracted to the cell. So the positive charge on the basic dye is going to interact with negative charges on the cell. You notice that the positive dye can go inside and neutralize the negative charges. So here so the dye has penetrated and is now inside the cell. The same thing with an acidic dye. The acidic dye is negatively charged. It interacts with positive charges in the cell. It enters the cell and neutralizes those positive charges. There is another type of dye that does not enter the cell, and since it does not enter the cell, it is not interacting, it is not going to neutralize the other charges. So here, this highly negative acidic dye is too big. It can't enter, so it stays on the outside of the cell. It doesn't neutralize any charges on the inside. It stays on the outside. This type of stain, since it stays on the outside, is called a negative stain. And don't mix that up with its charge. A negative stain stains the background. A negative stain is a background stain. So when you have a background stain, the organism is not stained, but the background is. So it's kind of like a negative image. Yes.
Is that the fluorescent type of?
Fluorescent?
Yeah, that you would see when you.
It's not fluorescent. We'll talk about fluorescent stains later. So the negative stain or the background stain, same thing, will stain the background. So the background will be colored and the organism will not. This is a really good stain if you want to look at the true shape of the organism, because in these stains you don't heat fix the slide, and when you don't heat fix the slide, you don't damage the cell. There's always some damage goes on with heat, you know. So if you don't damage the cell, you see the true shape of the bacterium. Its natural shape. And also, you can see structures on the outside of the cell that you might not be able to see with normal staining. And these would be called like a glycocalyx or capsule, which is kind of like a jello capsule around the outside of the cell. Those are destroyed with heat fixing, and you don't see them. But what you'll see with a negative stain is you'll see the bacterium, and you'll see like this halo around it, which indicates an area of no staining. Well, that area of no staining is there because that capsule is surrounding the bacterium. So here are some pictures of different stains. A simple stain where everything is the same color. Don't pay attention to the green. Everything, all the bacteria are the same. Differential stain where some bacteria, like the gram stain, where some, you can see in this case gram negative, the red, and gram positive, the purple or blue. Here's a background stain, India ink, and you can see the organism is unstained as well as an area around it, okay, which means the presence of a capsule. Methylene blue, which is a simple stain. Here's another differential stain. It's the acid-fast stain. Acid-fast stain uses multiple components. In this case, the red cells are the cells that are acid-fast. Don't get this mixed up with the gram stain. The components are different, and they're put on in a different order. The acid-fast stain is a very special, is a very particular stain you use for looking for certain disease-causing organisms, and there's only a couple. Tuberculosis, and the bacteria that cause leprosy, and a bacterium, I can't remember the other one. But, bacteria in the genus Nocardia, I don't know what disease they cause, and also bacteria in the genus Mycobacterium, which causes leprosy and TB. So this is a really good stain for identifying if a person has TB or not. You know, if they have, you take a sputum sample and you just stain it directly, and you look for the presence of these red rods. And the presence of red rods in the acid-fast stain means the presence of tuberculosis. Here's a flagellar stain, a special stain, and you can see the flagella around the rod, those little light structures. Here's another differential stain, a spore stain. In a spore stain, all of the vegetative cells, all of the normal cells will be red, and all of the spores will be blue. So inside you can see the presence of little blue cells, little blue structures. Those are spores. But you really got to look for the presence of the blue inside of a red cell. Then you can be sure that it has spores. Not all bacteria produce spores, very few. Not very few, a significant amount, but in general, most of them don't. So the gram stain procedure, you apply, you first of all, you make a thin smear, heat fix it, apply crystal violet for 1 minute, wash it off, apply iodine, wash it off. The iodine is a mordant. The iodine makes the crystal violet stick more to the cell wall because of cross-linking. What is cross-linking?
Exchanging, I just think like genes a little bit, but.
Here are two fingers. Cross-link them. Well, you hook them together like that. So the iodine is a cross-linking agent that hooks two things together. So what are you doing when you hook two things together? You're making it bigger. You're making it more complex. So when a dye seeps into the cell and you cross-link, it's harder to get out. Unless if you're gram-negative, which that means you have a really thin cell wall and it's easier to get out. Gram-positive, thick cell wall, harder to get out. So after the alcohol wash, anything that's gram-negative is going to decolorize, lose the dye. And it will pick up the safranin when you hit it with the safranin in the last step.
I have a question.
Mm-hmm.
Why do we heat fix it if we know that it's going to damage the cell?
Well, it's kind of tricky. You don't want to damage it too much. You want it to keep its natural shape as much as possible, but you don't want it to wash off the slide. So if you don't heat fix, then it won't stick to the glass, and you'll lose it. So it's kind of like the worser of two evils. Worser? Worse. Here's an acid-fast mycobacterium samples and a sputum sample. This person is very sick.
So the only thing that differentiates between gram-negative and gram-positive is the dye that it picks up, or is there some structural feature that distinguishes gram-negative bacteria from gram-positive?
There's some structural differences, which we'll learn later. They're important. Seen this negative staining, endospore staining, flagella stains. This just talks about the different dyes. You don't have to memorize these dyes. You'll become accustomed to them when you work with them in the laboratory. Because you work with crystal violet already. Methylene blue you haven't, but it's a simple stain. We're probably not going to be working with it. Safranin, you're working with it already. Malachite green, that's going to be used in spore staining. Nigrosin or India ink, one of these two we'll be using in background staining. Simple stains, just one stain, anything with one stain. The gram stain, the acid fast, the spore stain. We won't be doing the acid fast stain, but we've done the gram stain and we will do the spore stain. Those are differential stains. Now get the hang of that. What's a differential stain? Allows you to see a difference in structure. We're not going to be doing any special stains like, well, we won't be doing flagella stains, but we will be doing a spore stain.
Yada, yada, yada. Okay. These slides that have a lot of words on them, these are good to just kind of sum up things that you've been studying. Okay, so look at these and make sure you understand everything that's there, but it's nothing new. Okay, now what about microscopy? Light microscopy uses a compound microscope. That's what we're using. Compound because why? I got time, so. Why compound? Hmm? Where? Where are they? Yeah, we didn't talk about the ocular lens, but you have the objective lens and the ocular lens. The power on the ocular lens is 10x. The power on the objective is 10, 40, or 100. I think a few of you have a 4 in there too. So if you're using a 100 objective with a 10x ocular, what's the total magnification? 1,000. So it's the product of the ocular and the objective. So here it is. Okay. These all have different names. What are the 3 ways you can adjust the quality of light? Well, one, at the source, the intensity control. This is a little bit different from ours. The iris diaphragm, right there. And this whole unit right here, which is the substage condenser. The substage condenser is like a focusing lens for the light. Why do you want to focus the light on your object that you're looking at? To make it brighter, to make it more intense. Here's the ocular, of course, light intensity, okay. So here's the source right there. It comes up through the substage condenser, which focuses the light on the specimen, which goes into the objective lens, hits another lens. The prism bounces it to the ocular lens. So this is the path of light. The objective lens power times the ocular lens power is the total magnification. What's resolution? Resolution is, if you just think about it, it's how well you can see something. It's not a matter of focus, because you can have something in focus and still not be able to see it very clearly. One way to think about resolution is whether or not you can see 2 points next to each other clearly and distinctly. Let's say you have 2 points right in front of you on a piece of paper. You can see them perfectly as being separate and distinct. But the further away you put that piece of paper, the more chance there is of you not seeing them as 2 separate points. They'll blur together into 1 point. It's in focus, but you just can't make out whether or not they're 2. No. So whether or not you can see 2 points as being separate and distinct really has to do with the quality of lenses that you're using. The better the lens, the better the resolution. So the resolving power, which is a measure of resolution, is equal to the wavelength of light that you're using, lambda, divided by 2 times the numerical aperture. And the numerical aperture is a number that is stamped on the objective lens. If you go look at the lens next time, you'll see a number, you'll see NA number. And that is a quality of the manufacturing process. So how can you improve a lens when you're making it? And this is something, do you remember the Hubble, do you remember when the Hubble first went up? Do you know what the Hubble is? You know, that big telescope in the sky? When it first went up, it didn't work right because they made it wrong. You know, it's like sending something to Mars and having it crash on the surface. You ever heard of that kind of stuff going on? Happens all the time. You guys read the paper? The telescope was made wrong because the mirror, which is parabolic, wasn't perfectly spherical. You know how things have to be perfectly spherical to bounce light? You know, at the inside of the sphere, at the inside of this parabolic mirror, it was fine. It would focus the light rays on the right point. But out here, where the mirror bent out, it wasn't completely in line with it should be a nice arc, and it wasn't. It was flatter over here than it was over here, so the light rays were focused somewhere else. And all those light rays should be focusing on one focal point. The problem was now the focal point was spread out all over the place. The same thing here. The quality of lens. The more money you put into the manufacturing process, the finer you can get that focal point. The more spherical you can make that lens, the more perfect you can make it. So the more expensive lenses have a larger numerical aperture. And what happens when this number, what happens when the numerical aperture is large here in this equation? What happens to RP? I can't hear you.
It increases?
No, look at the quotient here, lambda over 2NA. What happens when NA gets big? What happens to RP? You guys math majors? It's a good thing you're not. Yes, it gets small. So the bigger NA gets, the smaller RP gets. What's another way to make RP smaller? Lambda. Make lambda smaller. Right, so make lambda smaller or make NA bigger. What's lambda? The wavelength of light. How do we make that smaller? You can't really, not in a light microscope. You're stuck with the wavelength. But what if you used another kind of microscope that used a different kind of wavelength? Like what? What kind of microscope? What kind of microscope allows you to see things that are really, really small?
An expensive one.
The expensive one. Yes, but that's the numerical aperture function. Electron. So the wavelength of electrons, instead of being in what's the wavelength of light in the visual light? What? I can't hear you.
Saying 7 or 7.5.
7 or 7.5 what? Lambda, no. Nanometers. It's about 600 to 700 nanometers. That's light. That's visible light. What's electron wavelength? How many nanometers? You know what a wavelength is? Maybe we ought to back up. Wavelength, light travels like in a wave, right? And the wavelength is the distance from one wave to the next. Wave length. Makes sense. So in white light, it's 600 nanometers. How big is a nanometer?
Really, really small.
10 to the minus 9th meters. Here's a meter. So 1/billionth of a meter. It's kind of small. So 600 of those is the wavelength of white light, visible light, and about 10 of those is the wavelength of
the electron wave. So what happens to the resolving power? The value of the number, RP. It goes way down. So does that mean it's harder to see? No, it means it's better to see. So what's RP a measure of? The smaller the RP, the clearer the image. Because RP is a distance. It's a distance measure. And remember those 2 points that I was talking about earlier? It's the distance between those 2 points that you can see as being clear and distinct. So that distance gets small with increased resolving power. Yeah.
What's the smallest you can get RP to be? Like, what's the finest?
Oh, it's in your book someplace. It's like, I think with a light microscope, it's like, it's like 0.1 micrometers. Yeah, like you know what that is. What is that? What is that? 0.1 micrometers is 10 to the 10 to minus 7th meters.
So that means our microscopes are pretty bad.
Well, it's pretty good. I mean, it's, 10 to the minus 7, it's not bad. It's better than anything you got at home. But electron microscope would be like 10 to the minus 9th, something like that. 10 to the minus 8th, no, 10 to the minus 9th, 10 to the minus 10th, something like that. So it's a lot more powerful. The resolving power, the resolution is a lot more powerful. The distance, the RP, is a lot smaller. The smaller the RP, the more powerful is the resolving power. It's an inverse relationship. Yeah, okay, so this says it all. Notice that the magnification has nothing to do with it. Okay, it has nothing to do with magnification. So when I ask you on a test, how do you increase the resolving power? And I give you multiple choice and I say, increase the magnification, and you check yes, you'll get it wrong. Okay, you increase the resolving power by increasing the numerical aperture or decreasing lambda. Here, and this is the last thing we'll talk about today. Here's why we have to use oil. You ever look at something in the water and you go to grab it and it's not there? Why? Because the light rays are being bent. The light rays from the object coming out of the water, when they hit the surface of the water to the air, they get bent. So it's not a straight line to the object. So we call that, what do we call that? Refraction. Refraction?
Yes, refraction when the light is bent.
Yeah, refraction. Thank you. So that's what's happening when the same thing happens when light rays pass through air and glass. You get refraction. So it's when light rays pass through any two media that are different. There is a refractive index, and the refractive index tells you how much it's bent. So what happens is the light rays are getting, this is a sucky slide. It doesn't really explain it very well. Essentially what happens is when the light rays go through the glass and the air, they get bent. Okay, and some of them make it into the objective and some do not. Don't follow this. This one should be bent out of the way. These should be straight. So some of the light rays don't make it into the objective lens because they get bent out of the way because of the refraction. And this only happens when the objective lens is really, really close to the object you're looking at. And the objective lens is really, really small in the opening. So it's easy for those light rays to get bent just enough so they don't make it into the objective. Well,
When you put oil in that space, what happens is the refractive index of oil and glass is 0. So there's no light bending. So more light is captured by the objective lens when you use oil in that space.
Could you do the same thing with water?
N-
Could water work with instead of?
Well, it'll definitely have an effect. I don't know what it would be, though. I don't know what the cumulative refractive index is. You have to look up the refractive index of water and glass and see what they equal. What?
You should try it.
Oh, yeah. All right, yeah, next time, why not? Just don't tell anybody here you put water on their microscopes. All right, that's it.